Okay, so I think with this group we don't have to go into too many details about what emerald ash borer is. I think we're all pretty familiar with it. I just wanted to show this map to point out one of the salient features of this infestation and invasion has been the disjunct nature of the infestations, that we have the anthropogenic movement of the beetle into areas where we would not necessarily expect it, big jumps between infestations, so it's difficult to predict where it's going to be. And one of the ongoing issues with this beetle has been how hard it is to find before it's very well established. And when the beetle first came, there weren't very well established techniques for trying to locate buprestids. We did a lot of roaming around looking for D-shaped exit holes, the world's least effective survey technique, but there's been a lot of progress, obviously, since 2002. And the major technique that's been used, of course, is the purple prism trap, which is basically a big piece of purple flypaper, and I assume most of you who are listening are very familiar with this trap, but it acts as an attractant. They like the purple. There's also a caramone in there, the phoebe and manuka oils, which mimic the smell of a dying ash tree, which is what the emerald ash borer likes to get. And this has been widely used, thousands of traps out every summer, and it has proven to be a very effective detection tool. However, again, it's very difficult to locate this beetle at low densities, and it's often the case that the detection doesn't happen until the beetle is pretty well established in an area, and that's been an ongoing issue with all of our survey techniques. Another technique that's been used widely, and I know over in eastern New York State you've been using it a lot, up in the Saugerties area, and we're using it here in Connecticut now, is the woodpecker damage. And you can see a couple pictures here. These are trees from Connecticut. This one is over here in Waterbury. This one was in Southbury. This was just this spring. And this is showing you that woodpeckers really like the tree because it's full of tasty EAB larvae. However, again, this doesn't work until the tree is fairly heavily infested because the woodpeckers aren't interested until then. We've been working with another option, and this technique, this is Phil Careless up here. He's a master's student, or was a master's student, up at University of Guelph in Ontario, and his advisor is the one who had the idea, and he's the one who ran with it. So I wanted to acknowledge him right up at the beginning of this. So, you know, one of the big problems is that we have a green needle in a green haystack. This little green beetle is up at the tops of the canopies of these trees. It's up in the leaves. It's very small. The canopies are very tall. How do we find it? Well, we have this native wasp, and here's a picture of her, and she provisions her brood with adult beetles in the same family as emerald ash borer for food. So she uses these jewel beetles as baby food. And basically what she does is she goes from her nest up into the trees, collects the beetle, stings it, paralyzes it, and brings it back with her to her colony. So this is a free service, and we just have to figure out how to use it. And I really like this picture. This is a picture that Phil took. Here's a female. She's returning to her nest with a mating pair of emerald ash borers. So that's 2 at 1 blow. So let me introduce you a little bit more to our beetle, Ciceris fumipennis, otherwise known as the Smokey winged beetle bandit. This is the stage of her life cycle that we are most interested in. This is the adult female wasps. They are solitary digging wasps, and she does hunting. And she starts hunting here in Connecticut. We just started about 2 days ago. They emerge in the summer, and then she immediately starts hunting and looking for beetles. She brings them back to her nest, which is in compacted sandy soil, and you can see it's a simple tunnel with a little tumulus of dirt around the top, and then she builds in sequence these little brood cells, and each one contains a single larva. So she fills it full of paralyzed adult buprestid beetles, and when there's enough to sustain, to complete the development of a larva, she lays an egg on it, she seals the cell up, and she moves on to the next. And she'll do as many of these as she can over the course of the summer. And so when the egg hatches, it's in a little chamber full of all the food it needs to complete its larval development. It feeds, it matures, and then it overwinters as a larva inside a cocoon underground, and then they emerge in summer. So in the north here, we have 1 cycle per year. Down in Florida, they have 2. And the males are just around, you know, they are about half the size of the females, and they're still out now, but they'll be gone in a couple weeks. They mate and die, so they don't have anything to do with it. So this is the stage we're really interested in. And here you can see her peeking out of her hole. They have these distinctive 3 dots on their face, and it's very nice. It makes them very easy to spot when you're looking in the hole to see if you have the right thing. You just look to see if those little dots are looking at you. Here's just some more pictures of her coming up out of the hole. This is with the native wood-boring buprestid. Well, they're all wood-boring. Native buprestid, this is a Dicerca lurida. And here you can see her nice mandibles that she uses to carry the prey. One of the bonus things about this wasp is that she's very good at stinging buprestids, but she does not sting people. So that makes it easy to work with. And here she is again poking out. And you can see the holes here. So they have a very distinctive round, very round hole. They are generalists within the family of Buprestidae. So they'll take any jewel beetle they can find up in the tree canopies. And this is just some pictures of a range of Buprestids, all of them on this slide except for the emerald ash borer here are native beetles. And these are ones that we regularly get here in Connecticut. These are common native species. And as you can see, they really range pretty widely in size, shape, color, but as long as it's a buprestid, she'll take it. So if emerald ash borer is around, she's also getting emerald ash borer. We're very lucky in Connecticut. We have a really dense coverage of Ciceris colonies, and these are native colonies, occurred on their own. We didn't have anything to do with it. These are the colonies that we know where they are and have been monitoring. I'm sure there's more colonies out there, but these are the ones that we've been working with so far in the biosurveillance program. And also on this map you can see the green towns, the townships in green here are the towns in which we have detected emerald ash borer. The yellow are the counties that are under the quarantine. So currently, you know, as of this summer, we have purple traps in this part of the state and not this part of the state. And in Connecticut, things work on the town level because we're so small. Everything happens on a town level. So even though emerald ash borer is known to be in, say, West Haven here, we're not sure. It's probably in Milford, say, but we don't know for sure. And so Milford is not going to pay any attention to us until we find it there. And that's just how things work in Connecticut. So we still are trying to go ahead and find them in each town that we can. So this is a picture of a typical colony of Ciceris wompennis of the wasp. They like compacted sandy soil. They like a lot of sun. They like a little scrubby vegetation. It turns out that town baseball parks, elementary school baseball parks are the best thing for them, the fields. And they tend to be in the infield here. And you can see up here on the little inset picture, you can see some of the holes. And this is a little bit later on in the summer. And this picture here happens to be of the first colony that we found emerald ash borer at in Connecticut. This is in Prospect, Connecticut. And so these are 2 folks. They're doing surveillance. And so what we do when to do the biosurveillance is we have an active colony. The females each have their own burrow, but they do tend to have aggregations. They're not colonial in the sense of honeybees, but they do have aggregations. And so each female is coming out of her own nest and coming back to her own nest with beetles. And so when we do surveillance, we come on, we look around on the ground for any dropped beetles. They do tend to drop beetles because there's kleptoparasites that try to get eggs on the beetles before they bring them down into the holes. So the females are pretty ready to abandon a beetle if they think something like that might have happened. So we find drops on the ground and collect them up. And then the other half of the surveillance is sort of the hunting/fishing part of the equation, and that's with nets. And as the females are coming back to their nests from foraging, from hunting, we catch them in the nets. The females drop the prey, and they fly out of the nets, and the beetle remains. So when we're doing these sites, the question is, well, how many beetles do we need to catch per site to make sure, or to have a reasonable assumption that there's not emerald ash borers around or any of the other target insects that we're looking for? And you have a compromise between the amount of time you can spend at a site and the number of beetles, of species that you can get. And this is a simple curve. This is data from 2012 in Connecticut. Each of the dots represents one of our colonies. Each of the, so on our x-axis, we have the number of beetles caught at that site, and on the y-axis, we have the number of species that we got at that site. And obviously, the curve, the number of species increases rapidly with the number of beetles caught, but then it starts to level off as you're getting repeats of the same species. And the compromise level between the number of beetles to catch that's reasonable and the number of species, new species that you're going to get, is around 50 beetles, and that's what our goal is. Obviously, as you keep collecting beetles, you get more rarer species, but we're trying to maximize the number of places we can get to as well as the rarity. And of course, when you're thinking about the rarity of species, part of that is because any survey technique you're doing like this, you're going to have this kind of an issue where the species, there's going to be some species that you get a lot of, the really common species, and then you get this real typical Poisson distribution as you're going out, where you get one or two of the rare ones, and you have a very long tail. And if your Planopenis is if your EAB is a very small portion of what's out there, you're not going to catch it until it's really out there on the tail. So the sensitivity of the survey is going to depend to a certain extent on how many beetles you're catching at a site. But this is some, again, data from Connecticut from 2010 to 2012. And these two here, these two guys are the ones we get most commonly. These are the Lorida and Sexignata. They're both deadwood feeders, so we get a lot of those every year. Overall, we've gotten about 55 different species of Buprested in Connecticut. So we have a lot of colonies. We can't do them all ourselves, so we've been working with volunteers, citizen scientists. Sounds like you guys are doing a lot of amazing work with citizen scientists. This is one of our groups. This is the Wasp Watchers, and volunteers from all sorts of different walks of life. And Master Gardeners in particular have been a really important group. We have the Master Gardeners have this as an official outreach activity that they can do. That has really increased the number of people who have been able to do this. But for the Wasp Watchers, what we do is we assign them a colony that's near to their house. Fortunately, we can do that. We can usually get a colony within 10 or 15 minutes of their house. We provide them with equipment, with data sheets, instructions, some reference guides, a map to their colonies, and nets, and then we give them a hands-on training, because that seems to really be important to help them understand what they're doing, and then they go back and monitor their colony. This program, we started it in 2010, and as you can see over the years, it has been increasing, the number of watchers we have, the number of people who are coming back, as well as the number of colonies monitored by those watchers and the number of beetles collected. And of course, as we've been getting better at this, we're also increasing the number of colonies and beetles that we've been catching. So last summer, we collected 3,600 Buprestids, and the watchers collected about half of those. So it's really been a very successful program. This summer we're gearing up. We have about 80 people signed up. There'll be some attrition, but you guys have all worked with volunteer groups, you know that. But we have a lot of returning watchers, and it's very, very exciting. just wanted to give you a brief history of how biosurveillance has worked in Connecticut. Just wanted to put a picture shout out. This is one of our wasp watchers, John Kasankis, and he is actually the first person who caught emerald ash borer at this site in Prospect. This is my assistant, Milata Scott. She caught one too. And here is an emerald ash borer just hanging out on the ground. So with this slide, what I want to stress is that biosurveillance using these wasps for us in Connecticut has been part of our toolkit for detection of emerald ash borer. There's lots of different detection methods that we have. You know, the purple traps have been an important part of the toolkit. Delimitation surveys, we did a big delimitation survey in spring of 2013, and of course visual surveys, and this can be by arborists, by us looking for woodpeckers, homeowners calling in. So there's a variety of techniques, and I'll just sort of step through. And again, here's our map of the various towns, and the pink towns are towns in which we have detected emerald ash borer using biosurveillance. In some of those towns, it wasn't the first way we did it. But this was our first detection here in 2012. It was rapidly followed by detections on traps and another wasp colony. In the fall, we had a homeowner report. In 2013, our forestry state foresters led off a delimitation survey under the guidance of Nate Siegert. The Forest Service up in New Hampshire and a lot of other agencies collaborated. And we found some additional towns with emerald ash borer, and we were beginning to see that this infestation was a little bit larger than we'd originally hoped. And then in the summer, last summer in 2013, we found additional towns and ooched into our surrounding counties. So now the entire western half of Connecticut is under quarantine, and I'm sure it's over, you know, the rest of the state's probably going to go by the end of the summer. And these were additional finds this spring. And finally, this one was a catch yesterday, and with the biosurveillance with the wasp in West Haven, Connecticut. So that was the way to start the season off with a bang. But we're going to be, right now we're in trainings. Things are just going up, so we'll see what we happen this summer. One additional point, I don't know how much time I have left, but one additional point about this survey technique is that even though we're mostly thinking about emerald ash borer at this point, because it is a generalist within the buprestid family, any other potential buprestid invaders, anything else, any other agrillus species or other species within that family that might be an invasive can be monitored using these wasps because they're just looking for any buprestid. And this is just some of the species that we're concerned about. And actually our CAPS money, the Cooperative Agriculture Pest Survey, the CAPS money that we're getting is targeted towards Bigutatis, Phosocolus, and Coxalis. These are all oak, pests of oak that we're looking for, as well as the emerald ash borer. And with that, I'd just like to acknowledge our funding from CAPS and from Forest Service. We've gotten a lot of help and encouragement. Again, Phil Careless and all the other researchers I've worked with, and I guess that's what I have to say, and I'd be happy to answer any questions if there's time.